All right, we are now recording. So I missed the first couple of announcements, but that's all right. Right, so we are recording the talk today. We will post it on our YouTube page, and you can find that along with our previous talks. The short address to navigate there is tinyurl.com/cryo-EM talks. On that page, cryo-EMcenters.org, you can also find registration links for the future talks, including our talk on April 29th. We have the registration available. We're still confirming our speaker, but we can promise it will be another great talk. So you can register now and we'll be announcing the speaker in the coming days. Quickly, along with information about this seminar series on that website, cryo-EMcenters.org, you can find information about all of the transformative high-resolution cryo-EM program resources, including the newly established cryo-electron tomography centers and the fantastic training materials being developed by our curriculum development partners. So I have a quick screenshot of what that website looks like. You can find information about all the different resources, and we hope this website helps you utilize the resources that are being built to help the biomedical research community use cryo-EM to advance your research programs. But back to today's agenda. We do have with us here today representatives from each of the cryo-EM centers. Michael Schmidt, one of the principal investigators at S2C2, is here with us. Ed Eng, the manager at NCCAT, and Lauren Halesbeck, the project coordinator at PNCC. And they're each going to give a very quick update from their center. And then Mike is going to properly introduce today's speaker, Professor Richard Ebright, who's joining us today from Rutgers University. Final logistics, Mike will be moderating our Q&A. You aren't able to turn on your microphones to ask questions, so please use the Q&A function at the bottom of your window. We'll be looking in the Q&A, not the chat, so please use that Q&A to ask questions. If your questions are directed to logistics or access at the centers, our panelists here will answer them directly, and questions for Richard we will save to the end of his talk. And you can see questions that other people are asking. So if there's something particularly interesting or a question that you also have, feel free to upvote that. That will help us prioritize our discussion at the end of the talk. And so I'm going to turn over to the centers for the quick updates, and Ed is up first.
Oh, welcome, good afternoon, or good morning, depending where you're coming from. And I'm hailing from NCCAT here in New York City. We offer high-end instrumentation access, including access to one of our 4 dedicated cryo instruments, as well as a robust cross-training program that's right now remote, but we're going to shift to a hybrid model in the upcoming few months. We have an upcoming deadline for proposals April 1st, and if you want to find out more, you can go to nccat.nysbc.org. One other announcement, we are offering our tomography short course that was delayed from last year in a virtual format. The deadline for registration is Monday, so please do sign up and hope to see you soon.
Thank you, Ed. Lauren, you're up next.
Awesome, thank you. So one of the updates we have is PNCC only offers now 1 proposal type for all projects. That is our general access proposal. So this gives projects up to 480 hours a year and is valid for up to 2 years. And you can apply now using our new PNCC user portal that we just launched this last week. So that should show you all of your information for proposal tracking. And then if you do apply with our center, it takes about 40 days in order for your proposal to be reviewed in order to potentially receive a award letter. And then once approved, there's a recommended onboarding with myself. We also have had some microscope updates. So we have 1 Arctica with K3. We have 1 Krios with Falcon 3 K3, 2 Krios with Falcon 3 Biocontinuum, BioQuantum. And then we have our newest Krios, has Falcon 3 and BioContinuum. That was just happened this last week. In the future, we're hoping to outfit one of those Krioses with Falcon 4 and ColdFeg, updating all of them with fringe-free by 2021. We have a VitroBot, a Leica GP2, and we're still waiting on our VitroJet to be shipped. We're hoping it'll be shipped by the end of the month, and install is still being scheduled. We still offer both screening and data collection of single particle and tomography for all approved proposals. And we're currently under restrictions, only having people remote, hoping that we can have visitors on site again sometime soon.
Awesome. Thanks, Lauren. And now Mike.
Hi, I'm Mike Schmid. This is the background that you see behind me. You can see it all now. We have 3 Krios microscopes with K3s and Falcon 4s on them with energy filters, and access to 2 other microscopes, another Krios and a Talos for screening. We, as the other centers do, offer, of course, high-resolution data collection services and cross-training, virtual, and hopefully in the future, in person. We'd love to see you here. So that's our website. Just check us out, please. Thank you. Okay.
All right, I will stop sharing, and Mike, I'll let you introduce Richard.
Okay. So good time of day to you all, wherever you are. It's my pleasure to introduce Dr. Richard Ebright for today's cryo-EM Current Practices webinar. Dr. Ebright is Board of Governors Professor of Chemistry and Chemical Biology at Rutgers University and the Laboratory Director at the Waxman Institute of Microbiology. His research focuses on the structure, mechanism, and regulation of bacterial transcriptional complexes and on the development of inhibitors of these processes for anti-tuberculosis and antibacterial agents using structural biology, biophysics, and drug discovery. He received his AB and PhD degrees from Harvard University and performed graduate research at Harvard and the Institut Pasteur. In 1987, he joined Rutgers University, where he's been since, and as the director of that Waxman Institute, and he has been a Howard Hughes investigator from 1997 to 2013. The title of his talk today is Structural Basis of Transcription-Translation Coupling. Excuse me, Dr. Ebright.
Thank you, Michael. I trust you can see my shared screen.
Very well,
Yes.
thank you.
Okay, so the topic of my presentation is the structural basis of transcription-translation coupling. The work that I'll present today was a result of a collaboration between my laboratory at Rutgers University, the cryo-EM, cryo-ET facility run by Jason Kelber at Rutgers University, Gregor Blaha at the University of California, Riverside, and Min Su at the University of Michigan. Almost all of the results are the work of 2 outstanding young researchers in the lab, Chengyuan Wang and Vadim Mulatsov. Both of them are present and connected for this event, and any questions about methodology should be directed to them, presumably through the chat. The work that I'll be presenting was published in a paper in Science in September, and it's important to emphasize that very similar work with completely consistent conclusions was performed by Albert Weichselbaumer and his coworkers and published back-to-back in Science in September. So in bacteria, transcription and translation occur in the same cellular compartment and occur at the same time. In most bacteria, including the standard model organism, Escherichia coli, transcription and translation are coordinated processes in which the rate of transcription by a molecule of RNA polymerase, RNAP, synthesizing a messenger RNA is coordinated with the rate of translation by the first ribosome, the lead ribosome, translating the messenger RNA. The evidence for transcription-translation coupling in E. coli is compelling. Classic EM images, so-called Miller spreads of E. coli, show same compartment, same time, coordinated transcription and translation, with the RNA polymerase essentially always being in contact with the lead ribosome. You see here a representative Miller spread. You see the DNA of a gene running from left to right, and you see 1, 2, 3, 4, 5, 6, 7, 8 molecules of RNA polymerase transcribing that gene, synthesizing RNA of progressively greater length as the RNA molecules move through the gene. And those RNA molecules are being simultaneously transcribed. On each RNA molecule, you see dark density features corresponding to ribosomes translating the RNA as it is being transcribed. And most crucially, for each density feature assigned as RNA polymerase, there is a lead ribosome in direct contact with the RNA polymerase molecule. In short, in E. coli, RNA polymerase and the lead ribosome move in unison. In addition, transcription and translation in E. coli are kinetically coordinated. If one artificially increases the rate of transcription, one sees a corresponding increase in the rate of translation. Likewise, if one artificially increases the rate of translation, one sees a corresponding increase in the rate of transcription. In addition, transcription and translation in E. coli are functionally coordinated. A ribosome can rescue a paused RNA polymerase, pushing it forward, and an RNA polymerase can rescue a paused ribosome, pulling it forward. Transcription translation coupling in E. coli is mediated by transcription elongation factors of the NusG RFAH protein family. NusG serves as a universal coupling factor, carrying out coupling at essentially every gene in E. coli. The NusG homolog RFAH serves as a specialized regulon-specific coupling factor, carrying out coupling at a small subset of genes in E. coli that contain a specific DNA sequence required for RFAH to load onto RNA polymerase. NusG and RFAH each consists of 2 domains and a flexible linker. Each contains an N-terminal domain that interacts with the RNA polymerase and a C-terminal domain that interacts with ribosomal protein S10. NusG and RFAH each is thought to bridge RNA polymerase and the ribosome, with the N-terminal domain interacting with RNA polymerase and the C-terminal domain simultaneously interacting with ribosomal protein S10 inside the ribosome. Transcription translation coupling in E. coli also is thought to be modulated by a second transcription elongation factor, NusA. In 2017, Patrick Kramer and his coworkers reported a low-resolution cryo-EM structure of an E. coli transcription translation complex that they termed the expressome. They obtained this complex by halting a transcribing RNA polymerase and allowing a translating ribosome to collide with the halted RNA polymerase. But the messenger RNA molecule in the structure was not fully resolved, making it impossible to determine the length of the messenger RNA spacer between RNA polymerase and the ribosome active center. And the functional relevance of the structure has been challenged. It has been challenged because the structure was generated as a collision complex, not as a bona fide coupling complex. And it has been challenged because the structure is incompatible with simultaneous interaction of the NusG N-terminal domain with the RNA polymerase and the NusG C-terminal domain with the ribosome, and thus is incompatible with the predicted bridging of RNA polymerase and ribosome by NusG. Our objective was to define the structural basis of transcription translation coupling in E. coli. And our approach to do that was to obtain high-resolution structures of E. coli transcription translation complexes, which we termed TTCs, that contained defined length, known length messenger RNA spacers between RNA polymerase and the ribosome. And we obtained such structures both in the presence and in the absence of the coupling factors NusG and or NusA. For this work, we prepared and evaluated a set of 7 synthetic nucleic acid scaffolds. The sequences of the 7 synthetic nucleic acid scaffolds are shown at the bottom of the slide. Each scaffold contained 2 DNA oligonucleotides shown in black, and one RNA oligonucleotide selected from the set of 7 RNA oligonucleotides shown in red. Each scaffold contained DNA and messenger RNA determinants that direct formation of a transcription elongation complex, a TEC, upon addition of RNA polymerase. These determinants consist of a non-template strand DNA and template strand DNA with a 9 base pair non-complementary region corresponding to the unwound transcription bubble that exists inside the RNA polymerase active center cleft in a transcription elongation complex. In addition, these determinants consist of 14 nucleotides of RNA, identical in sequence in each of the 7 RNA oligonucleotides. These 14 nucleotides include 9 that are designed to base pair to the DNA template strand to form an RNA-DNA hybrid inside the RNA polymerase active center cleft in the transcription elongation complex, and 5 additional nucleotides of RNA that are designed to exit from the RNA polymerase active center cleft, passing out through the RNA polymerase RNA exit channel. The dashed box shows the boundaries of the transcription elongation complex that would be formed upon addition of RNA polymerase to these scaffolds. Each scaffold also contained a messenger RNA determinant, namely a translation start codon, AUG, that directs formation of a translation complex upon addition of a ribosome and an initiating tRNA. You see here the translation start codon, the AUG, in each of the 7 RNA oligonucleotides. The dashed lines indicate the position of that start codon, that translation start codon, within the ribosome active center P site, the peptidyl product site of the ribosome active center. Each scaffold also had a messenger RNA spacer between the determinants required for assembly of the transcription elongation complex and the determinants required for assembly of the translation complex. This messenger RNA spacer has a different length in each of the 7 oligonucleotides, RNA oligonucleotides, ranging from 4 or 5 or 6 or 7 or 8 or 9 or 10 codons in length. For each of these resulting scaffolds, we added RNA polymerase, the ribosome, the initiating tRNA, and optionally NusG and or NusA to assemble the transcription translation complex, the TTC. We then determined structures by use of single particle reconstruction, cryo-EM. We obtained 2 different structural classes, which we designate TTC-A and TTC-B. With nucleic acid scaffolds that have short messenger RNA spacers of 4, 5, 6, 7, or 8 codons, we obtained structures that matched the previously reported expressome, and we designate this structural class TTCA, shown here boxed at left. In contrast, with nucleic acid scaffolds that had long messenger RNA spacers of 8, 9, or 10 codons, we obtain structures of a new molecular assembly, a molecular assembly that has features that indicate that it is the molecular assembly that functionally mediates NusG dependent, NusA dependent transcription translation coupling in E. coli. And we call this structural class TTCB, and it's boxed at right. We obtain structures of TTCB in the presence of the coupling factor NusG, showing bridging by NusG, and we also obtained structures of TTCB in the presence of both transcription translation coupling factors NusG and NusA, showing coupling by both NusG and NusA. So I'll present TTCA first and then TTCB second. Here I show the structure of TTCA in the same view orientation as in the preceding slide, and at right in a second view orientation rotated by 90 degrees about the vertical axis. In TTCA, RNA polymerase appears at the top. The ribosome 30S subunit appears in yellow below. The ribosome 50S subunit appears in gray below. And 2 tRNAs, 2 transfer RNAs, are shown in green and orange, bound within the ribosome active center site. The messenger RNA is shown in red, 9 nucleotides at the 3' end of the messenger RNA form base pairs with the DNA template strand as an RNA-DNA hybrid inside the RNA polymerase active center cleft. Additional nucleotides pass out from the RNA polymerase active center cleft, passing through the RNA polymerase RNA exit channel. Messenger RNA then enters the messenger RNA entrance portal of the ribosome, passes through the 30S subunit, and reaches the ribosome active center, where the AUG at the 5' end of the messenger RNA base pairs to the initiating tRNA in the ribosome active center P site. We obtain structures of TTCA with nucleic acid scaffolds having messenger RNA spacers of 4, 5, 6, 7, or 8 codons. And the structure that's illustrated here has a spacer of 4 codons and a resolution of 3.7 angstroms. In structures of TTCA obtained with nucleic acid scaffolds having messenger RNA spacers of 4, 5, 6, 7, or 8 codons, the structural organization of the complex is the same. Thus, when superimposed, the relationship of RNA polymerase to the ribosome is the same. The differences in messenger RNA spacer length in these complexes containing messenger RNA spacers of 4, 5, 6, 7, or 8 codons are accommodated through differences in extensive compaction of messenger RNA in the RNA polymerase RNA exit channel and the RNA polymerase ribosome interface. Again, the messenger RNA is shown in red. The 3' end of the messenger RNA is engaging the RNA polymerase active center catalytic magnesium ion, shown as a sphere in purple. 9 nucleotides of messenger RNA are base paired to the DNA template strand as an RNA-DNA hybrid in the RNA polymerase active center cleft. Additional nucleotides pass out of RNA polymerase through the RNA polymerase RNA exit channel. Messenger RNA then passes across the RNA polymerase ribosome interface, enters the ribosome messenger RNA entry portal, passes through the ribosome 30S subunit, reaches the ribosome active center P site, where the translation start codon base pairs to the initiating tRNA in the RNA polymerase active center P site. As the length of the messenger RNA spacer is increased from 4 codons to 5 codons to 6 codons, the first additional codon and the first and second additional codon are accommodated by compacting messenger RNA in the RNA polymerase RNA exit channel here, and in the RNA polymerase RNA ribosome interface here. Thus, as one progresses from 4 codons to 5 codons, one additional codon is accommodated by bending the RNA, kinking the RNA in these regions. And as one progresses to 6 codons, 2 codons are accommodated by kinking the RNA twice in 2 opposite directions in these regions. As the messenger RNA spacer is increased from 6 codons to 7 codons and 8 codons, the additional RNA again is accommodated by compacting the messenger RNA in these same regions. In this case, the messenger RNA nucleotides become disordered, indicating that the compacted messenger RNA nucleotides adopt an ensemble of distinct conformations. The volume of the RNA polymerase RNA exit channel limits the maximum messenger RNA spacer length to 8 codons in TTCA. And this immediately explains our observation that TTCA can be formed only with nucleic acid scaffolds that have messenger RNA spacer lengths less than or equal to 8 codons. In TTCA, the RNA polymerase ribosome interface is extensive. There's approximately 4,000 square angstroms of buried surface area. And the interface involves contacts of the RNA polymerase beta prime zinc binding domain in pink, the RNA polymerase beta flap in cyan, and the RNA polymerase alpha 1 subunit in green. These modules interact respectively with ribosomal protein S4, ribosomal protein S3, and ribosomal protein S10, which are present and exposed on the surface of the ribosome 30S subunit. Our high-resolution structure of TTCA confirms that TTCA is incompatible with NusG bridging. We've obtained structures of TTCA both in the absence of NusG and in the presence of NusG. They are identical. When NusG is present, NusG N-terminal domain binds at its expected binding location on RNA polymerase, but that binding location is on the opposite face of the complex from the expected binding location of the NusG C-terminal domain. The two binding sites are more than 160 angstroms apart. They cannot be bridged by NusG. Thus, NusG cannot bridge within TTCA. Our structure of TTCA also indicates that it's incompatible with NusA binding. If one models NusA at its expected binding location relative to RNA polymerase, one observes extensive steric clash, essentially total steric clash with the ribosome in TTCA. So NusA also cannot bind to TTCA. Our structure of TTCA indicates that it also is incompatible with known aspects of transcription. Thus, TTCA is incompatible with formation of pause or termination RNA hairpins, which are important regulators of transcription, and is incompatible with binding of transcription anti-terminator Q21, another regulator of transcription. If one models these components at their expected binding sites relative to RNA polymerase, one sees extensive steric clash with the ribosome. Pause and terminator RNA hairpins and the anti-terminator Q21 cannot bind to TTCA. Indeed, in TTCA, even one of the subunits of RNA polymerase, the RNA polymerase omega subunit, is absent or disordered. One sees no density for RNA polymerase omega at its expected position. If one attempts to model omega at that position, one sees that there would be steric clash with the ribosome. In addition, our structures of TTCA show that TTCA is incompatible with known aspects of translation. In each ribosome translocation step in translation, the ribosome 30S head rotates by about 20 degrees relative to the ribosome 30S body. And this phenomenon is known as 30S head swiveling. In TTCA, the RNA polymerase ribosome interface spans both the 30S head, shown in brown, and the 30S body, shown in yellow. And because the interface in TTCA spans both the head and the body, formation of TTCA would be expected to prevent swiveling, trapping the complex in the unswiveled state and preventing formation of a swiveled state, and therefore preventing translocation and tRNA accommodation in translation. In summary, TTCA is incompatible with NusG bridging, it's incompatible with NusA binding, it's incompatible with known functional aspects of transcription, and it's incompatible with known functional aspects of translation. Taken together, these lines of evidence indicate that TTCA does not mediate general transcription-translation coupling. We infer that TTCA is an anomalous complex, a complex that's formed only when the messenger RNA spacer between RNA polymerase and ribosome is anomalously short. TTCA is a collisionome, or a crashome, or a collided expressome. It is not the expressome that functionally mediates general transcription-translation coupling. We turn now to TTCB. You see here the structure of TTCB shown in the same two view orientations shown previously for TTCA. We obtain structures of TTCB with nucleic acid scaffolds that contain messenger RNA spacers of 8, 9, or 10 codons. And the structure illustrated has a messenger RNA spacer of 9 codons and a resolution of 4.7 angstroms. If we compare the structure of TTCA at left and the structure of TTCB at right, we can see that the position of the transcription elongation complex, the position of RNA polymerase relative to the ribosome, is substantially different. The position of the RNA polymerase in TTCA is directly over the center of the ribosome 30S subunit. The position of RNA polymerase in TTCB is at the edge of the 30S subunit. There is a 70 angstrom translation that relates the position of RNA polymerase in TTCA at the center of the 30S subunit to the position of RNA polymerase in TTCB at the edge of the 30S subunit. There also is a 180 degree rotation of RNA polymerase between TTCA and TTCB. That rotation occurs about an axis that lies in the plane of the screen on the diagonal. Because of this difference in translation and rotation, the relationship of the messenger RNA to the ribosome active center differs in TTCA and TTCB. In TTCA, the RNA polymerase RNA exit channel is coupled directly to the ribosome mRNA entrance portal. RNA exits RNA polymerase here and proceeds directly into the ribosome. In contrast, in TTCB, the RNA polymerase RNA exit channel here is separated by about 70 angstroms from the ribosome mRNA entrance portal here. In TTCB, a 70 angstroms long, 12 nucleotide long messenger RNA segment, the segment highlighted in black, connects the RNA polymerase RNA exit channel to the ribosome mRNA entrance portal. This messenger RNA segment runs along the surface of the ribosome 30S head, interacting along its length through favorable electrostatic interactions with positively charged residues, shown as orange spheres, in ribosomal protein S3. The requirement for this additional 70 angstroms long, 12 nucleotide long messenger RNA segment, the segment highlighted in black, explains the fact that TTCB is obtained only with longer nucleic acid scaffolds, that have messenger RNA spacer lengths greater than or equal to 8 codons. Only such longer messenger RNA spacers are able to span this distance. In structures of TTCB obtained with nucleic acid scaffolds having messenger RNA spacers of 8, 9, or 10 codons, the structural organization is the same. Thus, when superimposed, the orientational relationship of RNA polymerase relative to the ribosome is the same. In TTCB, just as in TTCA, the differences in messenger RNA spacer length for different spacers are accommodated through differences in the extensive compaction of messenger RNA in the RNA polymerase RNA exit channel. You see here structures with messenger RNA spacers of 8, 9, or 10 codons. The differences in messenger RNA spacer length are accommodated by compacting RNA in this space, in the RNA polymerase RNA exit channel, and in each of these structures, nucleotides of RNA in the RNA polymerase RNA exit channel are disordered, indicating that they adopt an ensemble of distinct conformations. The volume of the RNA polymerase RNA exit channel limits the maximum messenger RNA spacer length in TTCB to 10 to 11 codons, which indicates that, and we have confirmed that this is the case, indicates that TTCB can be formed only with nucleic acid scaffolds having messenger RNA spacer lengths that are less than or equal to 10 to 11 codons. In TTCB, the RNA polymerase ribosome interface is small. There's just 200 square angstroms of buried surface area, and the interface involves contacts of the beta prime zinc binding domain in light pink with ribosomal protein S3. This very small RNA polymerase ribosome interface is supplemented by a large NusG ribosome interface. In TTCB, NusG bridges RNA polymerase in the ribosome. The NusG N-terminal domain binds to the RNA polymerase beta prime and beta subunits at its expected location. The NusG C-terminal domain simultaneously binds to ribosomal protein S10 located in the ribosome 30S head, its expected binding location. And the NusG linker connects the NusG N-terminal domain and the NusG C-terminal domain. In our maps, we observe density for the NusG linker, and in maps contoured at lower contour levels, we observe unambiguous density for each and every residue of the NusG linker. Thus, the structures indicate unequivocally that the proposed bridging of RNA polymerase and ribosome by NusG occurs. It occurs in TTCB. We also have been able to obtain evidence that NusG is functionally essential for TTCB formation. In parallel experiments in which we prepare and analyze complexes in either with or without NusG, we find that without NusG, no TTCB is obtained. So NusG bridges in TTCB and is essential for formation of TTCB. We also have obtained structures of TTCB in the presence of both of the proposed transcription-translation coupling factors, both NusA and NusG. In such structures, NusA binds to the face of RNA polymerase opposite NusG and forms a second bridge between RNA polymerase and the ribosome. We've obtained such structures with messenger RNA spacers of 8, 9, or 10 codons. The structure shown here has a spacer of 8 codons and a resolution of 3.5 angstroms. You'll see in the structure NusG in red, bridging RNA polymerase and ribosome in exactly the manner shown in the structures containing just NusG, and then NusA in light blue, bridging RNA polymerase and ribosome on the opposite face of the complex. We've also been able to show that NusA functionally contributes to TTCB formation. In parallel experiments in which we prepare and analyze complexes with or without NusA, we find that without NusA, substantially less TTCB is obtained. So TTCB also has coupling and bridging through NusA. In these structures, NusA forms a large, open, rectangular frame that connects RNA polymerase to the ribosome. This large, open, rectangular frame, this diamond-shaped feature shown in blue. 3 corners of the NusA open rectangular frame interact with RNA polymerase. One corner interacts with RNA polymerase alpha subunit C terminal domain 1. A second corner interacts with RNA polymerase alpha subunit C terminal domain 2. A third corner interacts with the RNA polymerase beta flap tip helix. And the fourth corner of the NusA open rectangular frame interacts with the ribosome here, interacting with ribosomal proteins S2 and S5, both located in the ribosome 30S body. This NusA open rectangular frame contains an internal flexible linkage, a known internal flexible linkage, the AR1, AR2 linker, marked with a blue circle, and it interacts with RNA polymerase through 3 flexibly linked modules of RNA polymerase, namely alpha subunit C terminal domain 1 and alpha subunit C terminal domain 2, which are connected to the rest of RNA polymerase through long flexible linkers. Linkers are denoted with black bars, and the RNA polymerase beta flap tip helix, which is connected to the rest of RNA polymerase through flexible connectors, indicated with the gray circle. The internal flexible linkage in NusA and the flexible connections between NusA and RNA polymerase enable NusA to maintain constant contact with the ribosome 30S body despite differences in the orientation of RNA polymerase relative to the ribosome 30S body. Indeed, in our maps, in our cryo-EM maps, we observe 3 distinct structural subclasses, which we designate B1, B2, and B3. The 3 subclasses differ in the orientation of RNA polymerase relative to the ribosome, differing by rotations of 10 to 30 degrees on each of 2 axes. These differences in orientation of RNA polymerase relative to the ribosome are accommodated by the flexing of NusA, this open rectangular frame, taking advantage of the flexible connection, the flexible linker within NusA and its flexible connections to RNA polymerase. This can be shown more clearly on a video, which starts with TTCB1 and shows the rotation of RNA polymerase and the flexing of NusA to yield TTCB, and then the further rotation of RNA polymerase relative to ribosome and flexing of NusA to yield TTCB. TTCB2, and the further rotation of RNA polymerase relative to the ribosome and flexing of NusA to return to TTCB1. We refer to the NusA flexible open rectangular frame as the coupling pantograph, analogizing it to an electric railway coupling pantograph. This feature, which is the flexible open rectangular frame that enables a locomotive to maintain constant contact with a power cable here, despite differences in orientation of the locomotive relative to the cable here. Thus, the NusA open rectangular frame, this blue diamond shaped feature, this flexible diamond shaped feature, enables NusA to maintain constant contact with the ribosome here, here, and here, despite differences in orientation of RNA polymerase relative to the ribosome here, here, and here, in much the same manner that this open rectangular frame, this flexible diamond-shaped feature, enables constant contact with the power cable despite differences in orientation of the locomotive. Our structures of TTCB indicate that TTCB is compatible with the formation of pause or termination hairpins. If one builds a pause or termination hairpin into the structure of TTCB at its expected binding location on the RNA polymerase, there is no steric clash. TTCB is compatible with binding of the transcription anti-terminator Q21 if one builds Q21 into the complex of into the structure of TTCB at its expected location relative to RNA polymerase. There's no steric clash, and all RNA polymerase subunits are present, ordered, and accounted for in TTCB. TTCB also is compatible with known aspects of translation. In TTCB, all RNA polymerase ribosome and NusG ribosome interactions involve the ribosome 30S head. Accordingly, and you can see that here, so accordingly, 30S head swiveling can be accommodated by simply rotating RNA polymerase and NusG with the 30S head. That is modeled in this video clip. One sees the rotation, the swiveling of the 30S head, the rotation of RNA polymerase with the swiveling and unswiveling. In NusA containing TTCB, NusA ribosome interactions involve the ribosome 30S head, 30S body here, and thus the entire interface spans both the 30S head and the 30S body. Nevertheless, exploiting the flexibility of the NusA coupling pantograph, 30S head swiveling can be accommodated by rotating RNA polymerase and NusG with 30S head and flexing NusA to accommodate. And that's shown in this model video clip with the RNA polymerase rotating with the 30S head as it swivels and unswivels, and NusA flexing and unflexing to accommodate that change. To summarize our results for TTCB, TTCB is compatible with and functionally stabilized by NusG bridging. It's compatible with and functionally stabilized by NusA bridging. It's compatible with known functional aspects of transcription. It's compatible with known functional aspects of translation. Taken together, these lines of evidence indicate that TTCB is the structural state that mediates NusG-dependent, NusA-dependent transcription-translation coupling in E. coli. In overall summary, our results provide high-resolution structures of the previously described expressome, namely TTCA. The results demonstrate the incompatibility of the previously described expressome with general transcription-translation coupling. The results provide high-resolution structures of a new structural state, TTCB, that has properties assignable to functional, general, NusG-dependent, NusA-dependent transcription-translation coupling. NusG stabilizes this new state, TTCB, by bridging RNA polymerase in the ribosome 30S head. NusA stabilizes this new state, TTCB, by bridging RNA polymerase in the ribosome 30S body. And NusA serves as a coupling pantograph that flexibly bridges RNA polymerase in the ribosome in a manner that allows rotation of RNA polymerase relative to the ribosome 30S body. In current work involving structural biology, we're performing a parallel analysis of transcription-translation coupling by the specialized NusG homolog RFAH. You may recall from the introduction that the NusG homolog RFAH carries out transcription-translation coupling at a small subset of genes in E. coli, namely genes that have a specific DNA sequence required for RFAH to load onto RNA polymerase. And we have obtained structures of a series of RFAH coupled transcription translation complexes, both with the RFAH alone and with the RFAH plus NusA, both with the specific DNA site required for loading and without the specific DNA site for loading, and at a series of different messenger RNA spacer lengths. These structures show that NusG and RFAH couple in essentially the same manner and confirm the notion that they are counterparts that bridge RNA polymerase and the ribosome, in one case at most genes, and in the other case at genes having a specific sequence for loading. In addition, we are carrying out an analysis of transcription translation coupling in the other domain of prokaryotes that carry out transcription translation coupling, namely the archaea. In current work involving molecular biology, we are determining whether the RNA polymerase ribosome, NusG ribosome, and NusA ribosome interactions that we see in our structures occur in living cells. And we are analyzing the functional consequences of disrupting those interactions for their effects on transcription in living cells. And we are analyzing the functional consequences of disrupting those interactions for their effects on translation in living cells. So I thank you for your attention, and would be happy to address questions of the system and to refer questions on methodology to the researchers who were first and second authors on the work, joint first authors on the work. So thank you.
Thank you, Richard. We do have a question on the analysis of the structures, and comes from Saba Sharad. You mentioned that RNA polymerase can rescue halted ribosomes and vice versa. Do you think the structures that you have shown us can shed light on this aspect?
The structures show that if you consider the two possibilities, when RNA polymerase is halted, a ribosome can resuscitate the RNA polymerase by pushing it forward. That pushing could be mediated by either of the coupling factors, NusG here in red, or NusA, but particularly by NusA. The NusA structural framework provides a coupling mode where a force applied by ribosome would be transmitted through NusA to RNA polymerase. So in principle, either of these coupling factors could provide for ribosome to assist forward movement of RNA polymerase, but NusA in particular. Conversely, an RNA polymerase molecule can assist the movement of a ribosome by pulling it forward. Again, in principle, either of these coupling factors, NusG or NusA, could accomplish this, could provide the coupling that enables the RNA polymerase to pull a ribosome forward. But NusG in particular has the structural features that conform to a tow rope, with one domain binding to RNA polymerase, one domain binding to the ribosome, a flexible connector between them. In essence, it is a tow rope that would allow RNA polymerase to pull a ribosome forward.
Mm-hmm. And maybe to follow up on that, in order to actually know or figure out whether a ribosome or the polymerase is stalled or not, and not coupled, there has to be a sensor. You know, you have to, those structures have to sense whether there is a force or some interaction between the ribosome and the RNA polymerase that's different from the steady state. And can you assign a sensor function to either of these two?
No, the sensor again could be mediated by either the sensor function could be mediated by either NusG or NusA. The sensor in this case simply is the coupling. If one considers, again, using a locomotive analogy or a locomotive metaphor, in a locomotive standard coupling, there are two components. There is a component that provides a forward force, and there is a component that provides a coupling through a tow rope-like connection. So a standard locomotive connection involves both a push bar and a chain. And so two components that match very closely, the push bar NusA and the chain NusG, the tow rope NusG. One simply needs a coupling that has both a relatively rigid component that can provide a forward force, and then a component that can provide a towing force, effectively a chain.
Another question. Are there known phenotypes associated with NusG and A mutants and deletions? Do you foresee that NusG and A mutations, deletions could deletion increases translation abortion?
So in normal E. coli strains, both NusG and NusA are essential for viability. So deletion of either of the two results in an inviable cell. One can engineer strains of E. coli in which deletions of NusG or NusA are tolerated, but even in those cases, they're tolerated poorly. So deletion of either of these components results in substantial changes in transcription and translation that are not tolerated in a wild-type cell.
Actually, Mike, do you mind if I pop in with a technical question?
Okay, go ahead, please.
You have so many absolutely beautiful structures. I'm just curious how much data goes into each of these. And it may be a long answer, but if you could speak to that a little bit, I would be very curious.
Okay, so here is a table of structures from our paper in September. And so these are NusG dependent and NusG plus NusA dependent transcription translation coupled complexes. And for those that show NCAT as the cryo-EM facility, data were collected on a cryos. For those that show Rutgers as the cryo-EM facility, data were collected on the Talos. So these are with a 300 kV instrument. The ones labeled Rutgers are on a 200 kV instrument. This column shows the particle numbers, and this the resolution. So this provides one framework for assessing that question. Here's a second table. This table shows the corresponding structures that we have obtained for the specialized NusG homolog RFAH, the NusG homolog that carries out transcription translation coupling only at a subset of genes. And here we've collected structures at each of the three national facilities. In each case, those would be structures on a cryos, and at Rutgers, those would be structures on a Talos. And again, some indication of particle numbers and corresponding resolutions. In most cases, each one of these structures represents 1 to 2 days of data collection, either on the cryos, the 300 kV instrument, or on the Talos, a 200 kV instrument.
Amazing, thank you. That perfectly answers my question.
And it shows a great use of all three centers, right? Actually.
It would not be possible without these three centers, and our best structures obviously come from for each structural class, our best structures come from data collected at one of the national centers on the 300 kV instrument.
I just want to pop in because we're coming up on the hour. So if people who have something else to run off to, thank you for joining us. We will stay here and field questions with Richard as long as he's willing to stick around with us. But for those of you who have to go, we will have the recording if you had a burning question you might miss. And we look forward to seeing you again. And I'll turn back over to Mike and Richard for more questions.
Okay, thank you. Another question, do you think there's coupling between transcription and different stages of translation? For instance, do initiation factors also take part in the coupling process?
We're trying to explore, in current work, we're trying to explore how the coupling first occurs and how the coupling decays, because we know there are circumstances under which coupling is lost. So we're very interested in these questions of what happens when the RNA polymerase and the ribosome are not yet close enough to form TTCB, and what happens when they move too far apart to form TTCB. And we've identified indications of how that occurs with an additional structural class that appears to be either the precursor to formation of TTCB or an intermediate in the loss of TTCB. So trying to relate the complexes that we observe to different stages of translation is something that's currently in progress, and then trying to relate all of the structural information to what occurs in the cellular context. Where and when in the cell does one see TTCA? Where does one see TTCB? Where does one see this new precursor to formation of TTCB? And obtaining a complete answer will require both structures and biochemical data.
Let's see, another question. Could there be any implications of the TTC mechanism on other factors such as EFTU and EFG?
That's another area that we are actively working in. Our collaborator, Gregor Blaha at University of California, Riverside, has provided us with the complete set of translation elongation factors and with amino-acylated tRNAs. And we are now attempting to solve structures of both TTCA and TTCB trapped at different stages along the translation amino acid addition cycle in order to see how these factors, how the transcription translation complexes impact the mechanism of translation. You've seen me show video clips of a model of RNA polymerase rotating with the 30S head of the ribosome during 30S head swiveling and unswiveling. We'd like to directly capture those swiveled and unswiveled states and to be able to confirm that reality corresponds to the videos that we were showing.
Okay, and one in terms of the construction of this whole complex, that all occurs after the 30S and 50S ribosome are together, or is this built before the complete ribosome, large and small subunits are together?
In these experiments, we take advantage of the fact that in a system that has just messenger RNA with an AUG translation start codon and an FMET tRNA, the initiator tRNA, one can assemble a translation complex just by adding a 70S ribosome, the intact ribosome with 30 and 50S subunits already pre-assembled. And so we use this shortcut to build the complex. We have no information on what would occur if we built the complex in the stepwise fashion that's required for assembling a translation complex from individual 30S and 50S subunits.
Okay, thank you. I don't see any more questions in the Q&A. If there are other questions, especially for the technical aspects, we saw a couple of them, but any questions are fair game. Type them in, and if we don't see in a couple of minutes, we'll thank you, Richard.
Okay, thank you.
Actually, I'll pop in with another technical question real quick since you're here. I'm curious about local resolution in your structures, and I'm guessing it varies a lot. But for example, your TTC-A versus B, is there like a huge difference in the range of local resolution?
So here would be a picture that would provide some insight to that. This is TTCA. So this is TTCA with NusG bound to it. And the ribosome has resolutions that are in the blue. The RNA polymerase shows much lower local resolution. And that is the consistent pattern for all of our structural classes. The overall FSC resolutions, in the case that you're seeing here, this is an overall FSC resolution of 3.7 angstroms. But that reflects excellent local resolution for the ribosome, poorer local resolution for the RNA polymerase. And that is, again, the consistent pattern. If we looked at, here we have a complex with this spacer length, we get primarily TTCA and only a little bit of TTCB. And then one can see that under these circumstances, TTCB has much lower resolution in the RNA polymerase region. When we look at TTCB complexes with spacers where we obtain exclusively TTCB, we go back to this standard pattern of good resolution for the ribosome, weaker resolution for RNA polymerase. Across the board, what we say. Here's with complexes having both NusG and NusA.
And that's consistent with the fact that the largest mass is the ribosome. If you did focused classification of the RNA polymerase part of it, would you rescue slightly better resolution in the RNA polymerase part? Just because that would be the focus of your alignment then in that case.
So if you look at panel A of the figure, which gives the data analysis workflow, a structure such as the one shown here, in panel E, is coming with a focused 3D classification on the RNA polymerase to obtain that information. So again, you're absolutely right. The reason the ribosome comes up first and easiest, that's most of our material, we find that first. In some cases, such as the structure of TTCA, one can go straight from the data to the structure with no focus classification. And in other cases, to obtain this result, we do need focus classification with masks looking only at the RNA polymerase.
Okay, very good. Okay, let's see.
All right, I think I'm going to go ahead and stop the recording since I don't see any other questions.